Extracellular vesicles, including exosomes, are membrane vesicles released from various cells. Those contain proteins, lipids, and nucleic acids. Many people have therefore been interested in them as intercellular messengers and disease markers. This isolation method is easier than conventional ones. It realizes the preparation of high purity and intact exosomes. Fujifilm Wako Pure Chemical Corporation has succeeded in the development of such a novel exosome isolation kit. This kit is based on the affinity purification method that is specific to phosphatidylserine localized on the surfaces of exosomes. It is composed of these 6 parts. The exosome isolation process is composed of these 4 steps. Prepare samples pre-treated from cell culture supernatant, serum, or plasma following the procedure. On magnetic beads, immobilize biotin-labeled exosome capture that can capture exosomes in samples. Wash 60 microliters of biotin capture magnetic beads with exosome capture immobilizing washing buffer. Spin down the magnetic beads and remove the buffer. Add another 500 microliters of exosome capture immobilizing washing buffer, and then 10 microliters of biotin-labeled exosome capture. Incubate the tube by mixing on the rotator at room temperature for 10 minutes. Remove the reaction solution. Wash the magnetic beads with exosome capture immobilizing washing buffer twice. The preparation of exosome capture immobilized magnetic beads has been completed. Incubate the sample solution with the exosome capture immobilized beads obtained in the previous process to have exosomes captured by the beads. Add exosome binding enhancer to the sample solution in advance. Add this solution to the exosome capture immobilized beads obtained in the previous process. Vortex the tube. Incubation the tube by mixing on the rotator for an hour or more. As the incubation of exosomes with the magnetic beads proceeds, the homogeneously suspended magnetic beads will become agglutinated. Spin down the magnetic beads and remove the reaction solution with a magnetic stand to obtain EV's binding beads. Wash the EV's binding beads obtained in the previous process, and elute captured exosomes from the beads to acquire purified exosomes. Add exosome binding enhancer to a needed volume of exosome capture immobilizing washing buffer in advance. Wash the EV's binding beads with the exosome capture immobilizing washing buffer. Spin down the magnetic beads again and completely remove residual washing buffer with the magnetic stand. Remove exosome capture immobilizing washing buffer from the tube as completely as possible, because the residual buffer may inhibit the subsequent elution process. Add 50 microliters of exosome elution buffer to the washed EV's binding beads. Vortex the tube. Tap the tube in the case of insufficient dispersion of magnetic beads. And leave it at room temperature for a minute for incubation. Spin down the magnetic beads. Attach the magnetic beads to the tube inner wall with the magnetic stand. Collect the reaction solution, including exosomes detached from the magnetic beads, into another tube. Add 50 microliters of exosome elution buffer to the magnetic beads again, and elute exosomes in the same way. Collect the reaction solution, including detached exosomes, into the same collection tube. In total, purified exosomes will have been collected. With the acquired purified exosome solution, the particle sizes and concentrations can be measured with particle analyzers such as Nanosite LM10. MagCapture Exosome Isolation Kit PS version 2 can easily isolate highly purified exosomes from cell culture medium, serum, and plasma. We hope that your research goes well with this kit.